Now this one second lecture about urea cycle. Up to this carbamyl phosphate I discussed in previous lecture how carbamyl phosphate is synthesized. Now carbamyl phosphate is condensed with this ornithine. This ornithine is condensed with this carbamyl phosphate and ultimately formation of this cytolin. Up to the cytolin synthesis, uh, urea is urea run up to this cytolin within the mitochondrial matrix. Urea occurs up to this cytolin synthesis within the mitochondrial matrix. So carbamyl phosphate is condensed with this ornithine and ultimately synthesis of cytolin. This step condensation is done by ornithine transcarbamylase. Ornithine transcarbamylase. So up to this occurs in the mitochondria. Now this cytolin come out from the mitochondria to the cytosol. Now this cytolin, this is the chemical structure of cytolin. You just follow the this colored atom like nitrogen, carbon in the previous chemical structure. Step by step, just this chemical structure is altered. Step by steps, this chemical structure is altered. Here, this is the ornithine chemical structure. Here, carbamyl phosphate is condensed, and ultimately we can get this chemical structure. Then it enters into the cytosol. Now, now this cytolin is now converted into arginosuccinate. Here you follow in the cytolin there is one nitrogen atom and in the arginosuccinate is double nitrogen atom. But how? We can get another nitrogen atom from the aspartate. Here you follow aspartate has one nitrogen atom and from this aspartate we can get this nitrogen atom and added to this cytolin and ultimately arginosuccinate is formed or arginosuccinate is synthesized. So we can say that arginosuccinate has double nitrogen atom. Here ATP is converted into AMP. It means energy is utilized in this step. ATP. So ATP is converted into AMP. And enzyme name of the enzyme is arginosuccinate synthetase. Now, arginosuccinate has double nitrogen and now arginosuccinate is converted into arginine. Arginosuccinate is converted into arginine. So, now arginosuccinate is converted into arginine. Here, from this arginosuccinate, fumarate is puffed out. From this arginosuccinate, fumarate is puffed out. This is the chemical structure of fumarate. So, this chemical structure is removed from this chemical structure. So, ultimately, we can get this chemical structure is arginine. If we if we follow carefully this chemical structure after removing this chemical structure, it means after removing the fumarate, we can get this chemical structure arginine. Now, from the arginine, we can get this ornithine by the arginase by the arginase. Actually, this enzyme arginase hydrolases. If you go through the living code another page, in the next page, you will find that arginase hydrolases enzyme. Name of this enzyme is arginase hydrolases. It means here water is added. Water is added to this chemical structure. Water is added to this chemical structure. It means water is added to this arginine. So by the help of arginase hydrolases enzyme, ultimately we can get this chemical structure. It means we can get urea and we can get this chemical structure. It means its name is ornithine. This ornithine again enters into the mitochondrial matrix and this recycling occur. That's why Euro cycle also called ornithine cycle. Why ornithine cycle is Euro cycle is called ornithine cycle? You already understand that ornithine recycling occur. That's why Euro cycle also called ornithine cycle. As after formation of the ornithine enters into the mitochondrial matrix, then ornithine and carbon